Research is the cornerstone of innovation in clinical practice, driving improvements in patient outcomes and shaping the future of medicine. Welcome to the Global Rhinology Network Topical Research Symposium. I'm Ashok Rokde from Winchester, UK, and it is my privilege to welcome you as a host. Joining me as co-hosts are Louise Melia from Bath, UK, and Professor Thibaut Van Zelle from Ghent, Belgium. This symposium showcases fascinating topics across 4 key modules: General Rhinology, COVID session, and Research in Chronic Rhinosinusitis. A special highlight is the PhD session featuring research conducted by enthusiastic young rhinologists who represent the future of our field. This symposium brings together leading global minds in rhinology who will highlight latest developments with a focus on translating research into impactful patient care. The Global Rhinology Network is dedicated to advancing knowledge innovation. Thank you for being part of this exciting journey. Thanks to Medtronic and Karl Storz for their generous support in making this symposium possible. Welcome to GRN-Topical Research Symposium.
Right, so we'll move on to our last speaker of the day. So it's Roshan Nepal, who's a scientist having graduated in biotechnology from Tribhuvan University in Nepal. He commenced his PhD in 2019 and has a keen interest in phage therapy, antimicrobial stewardship, and AMR research. And today he's going to talk about his PhD project, which is the role of prophages in Staph aureus virulence and implications for chronic rhinosinusitis.
Thank you for the introduction. Just let me share my screen. Can you see my screen?
Yes.
Okay. Yeah. Good evening, everyone. I'm Roshan Nepal, and I'm PhD candidate at University of Adelaide, Australia. And today I'm going to talk about my PhD research, which is about prophage biology. To briefly introduce about chronic rhinosinusitis, I don't think this audience needs any introduction about chronic rhinosinusitis. So it is an inflammatory disease of paranasal sinuses, and broadly it is categorized into 2 types, chronic rhinosinusitis with polyp and non-polyp. And there are many factors that affect pathophysiology of chronic rhinosinusitis, but among them, usually bacteria, and specifically Staphylococcus aureus is most often associated with CRS with polyp cases. So I wanted to work with Staphylococcus aureus and see how it impacts the disease severity in chronic rhinosinusitis. As you know, that Staphylococcus aureus is a gram-positive bacteria, which is almost carried by 30% of population as a commensal bacteria. So this is one of the most successful colonizer in humans. However, there is no any clear distinction between what is commensal and what is pathogen in terms of virulence factors and other genes in Staphylococcus aureus. So if you look into genetics of Staphylococcus aureus, and if you look into genetics of virulence in Staphylococcus aureus, most of the virulence factors are usually carried in mobile genetic elements, which are also called MGEs. Those mobile genetic elements are plasmids, transposons, and one of the most important mobile genetic element is also prophages, which is short sequences of DNA that is integrated into bacterial genome. Those sequences are usually mobile and carry virulence factors. So this is one of the important factor in genetics of Staphylococcus aureus. So to briefly give you an introduction about what is a prophase, like I said earlier, prophase is a short sequence of DNA that is completely or stably integrated into bacterial genome. So those are mobile elements inside the bacterial genome that comes from a bacteriophage, typically temperate bacteriophages. And these temperate bacteriophages or prophages that is integrated into bacterial genome often equips bacteria with multiple virulence factors, thereby increasing their fitness in the host. For example, heightened biofilm, heightened virulence factors, and other things. Um, Various researches has proven that bacteria with prophages or bacteria that has prophages inside it are more virulent in terms of disease severity. For example, in IBD, bacteria with prophages are more virulent. But however, role of prophages in Staphylococcus aureus and CRS has never been understood before. So this is why we wanted to look into prophages and its associated virulence factors in Staphylococcus aureus isolated from CRS patients. So our hypothesis was accessory prophase elements or accessory prophase sequences provide Staphylococcus aureus survival fitness and may impact CRS disease severity. For this, we had two aims. Our first aim was to investigate prophase diversity in Staphylococcus aureus isolated from CRS patients. And then after that, test if presence of certain prophages alter bacterial virulence or not. So to give you a picture of how we did this, we first collected Staphylococcus aureus from CRS patients, 28 from without polyp and 30 from with polyp patients, sequenced them, and using different bioinformatics tool, we analyzed the prophages inside bacteria and determined what kind of prophages and what kind of virulence factors these bacteria have. And moving on to results, what we found was prophase elements in CRS with polyp cases were usually higher, which means prophase elements in bacteria that were isolated from more severe case of CRS, which is CRS polyp, were higher. Polyp cases were higher, but this was not statistically significant. So we further looked into the state of prophages. Like prophages can be complete or intact, or they can be incomplete or incomplete prophages. So what we found was here, just let me use a pointer. Here you can see that there are more greens in here, which means the greens are the intact prophages, which are active prophages. So in CRS with polyp cases, the greens are many, which means in these Staphylococcus aureus bacteria, intact prophages were higher in number, and this was statistically significant. After finding this, we wanted to know what kind of prophages were those prophages. So we found that in case of chronic rhinosinusitis with polyp, Sa3int, which is also called HLB converting prophages, this type of prophages that had integrase type 3 were more prevalent. And these results are published, and if you want to see or learn more about these prophages, then read the paper. These are the papers that we published. So after that, after finding that Sa3int prophages were highly abundant in CRS with polyp cases, we thought that bacteriophages may play a role in virulence of bacteria, thereby impacting disease severity in CRS. So next, what we wanted to do was we wanted to look into more detail into Sa3int prophages, which are integrase type 3 prophages. And we already know that these prophages carry certain virulence factors like CHIPS, CHIPS, SAT, and SCIN that are known to evade the immune system of humans. So prophages or bacteria that carry these prophages are more able to evade the human immune system, so they can infect and cause chronic infections. So for our second approach, what we did was we isolated those kind of prophages from one bacteria, which is a donor bacteria, and we transduced or we inserted those prophages into another bacteria, and then created a lysogen and wanted to see if there is any transfer of virulence factors in those bacteria or not. And the first thing that we observed was deletion or downregulation of HLB gene, which we can see in these areas here. although all of the bacteria in this plate are Staphylococcus aureus, there is a SLB or beta hemolysin in these strains, and there is absence of beta hemolysin in these strains, which means these bacteria or these colonies have lost the beta hemolysin property because of the insertion of the prophages. And we confirm this with sequencing. In sequencing and multiple alignment here, you can see that the prophages has inserted in this area, which is a lysogen. So we can clearly say that the prophages are integrated inside HLB gene, and then they downregulate production of HLB. After that, we also wanted to know if there is any change in growth kinetics between those two strains, and we found that although the prophages was integrated inside the bacterial genome, there was no any growth, there was no alteration of any growth curve, or there was no any alternation in the doubling time of the bacteria. Both of the bacteria grew equally. Also, there was no any change in biofilm, although some of the research reports that there is increased biofilm in lysogens. We didn't see increase in biofilm significantly. So after that, what we did was we wanted to see the proteomics of the bacteria and the lysogens. So we did a GCMS, or we did proteomics analysis on the secretome of both of the bacteria. The bacteria that was genetically modified, the lysogen, and its recipient bacteria, and also the donor bacteria. So in this graph, in this heat map, what you can see is these bacteria are the recipient bacteria, and these are the donor bacteria, and these are the lysogens. These are the genetically modified lysogens. And what we can see, there is clearly upregulation of all the genes in group 1 and 3 in lysogens compared to the recipient bacteria. And all of the genes were also upregulated in the donor bacteria. So these are the genes that were carried by the Sa3int prophages. We can clearly see that the genes are upregulated in the lysogens, and these were carried by Sa3int prophages. In altogether, 22 genes were upregulated and 2 genes were downregulated in these lysogens. Further, we also wanted to see what kind of which genes were upregulated and downregulated in this volcano plot. We can clearly see that the gene of our concern, SAK, SCN, and CHS, which are a group of immune evasion cluster genes that help bacteria evade the human immune system, were upregulated in the lysogens. We can also further see the same data in a different way, where we can clearly see that chemotaxis inhibiting protein, or CSIPS, or staphylococcal complement inhibitor, SCIN, and staphylokinase, all of these genes which are responsible for immune evasion are upregulated in bacteria. And the most interesting part of this research is there are also other hypothetical proteins or hypothetical genes that are upregulated with function we are not able to elucidate with this research. So in conclusion, we can say that prophages are abundant in clinical strains, and they may have some role to play in disease, because lysogens always outcompete the normal flora in clinical patients. And also Sa3int prophage integration doesn't significantly alter the bacterial growth dynamics or biofilm formation, although there are some reports of heightened biofilm formation in a lysogen. And finally, we can say that bacteria infected with Sa3int prophages are more severe, or they carry factors that cause immune evasion in the exoprotein, and these proteins are secreted as an exoprotein by the bacteria. And finally, I would like to acknowledge the organizer for this symposium, and also my supervisor and all of my ENT research group. Thank you very much. I'm happy to take any questions.